Why do we sleep? Why do we dream? What do these processes do for our minds and our bodies? Welcome to the Sleep Science Podcast, where we discuss all these issues. I'm Penny Lewis, a sleep scientist and the presenter of this show. The Sleep Science Podcast is a production of the NAPS Sleep Lab at Cardiff University. You can find out more about our guests, the podcast itself, and our sleep research at Cardiff on our podcast webpage. My guests today are Professor Dan Margoliash from the University of Chicago and Dr. Tim Brawn, a behavioral neuroscientist from MIT. Dan has been working with birds and birdsong for much of his career. We'll talk about how baby birds learn to sing like their parents and why this provides a perfect model system for studying memory consolidation and sleep. We'll also talk about some of Tim's examinations of how grown-up birds learn to recognize new songs and how studying this reveals sleep's role in handling interference between similar songs and in restructuring well-learned memories through a phenomenon called reconsolidation. In case you're curious, the bird song you heard at the very start of the program is the song of the starling. A lot of your work on sleep has been in birds. In fact, a lot of your work in general is in birds. So the obvious question is, why would you ever look at sleep in birds? What's interesting about that?
The fact that it's important for birdsong learning and the perception of sounds, which are things that we had no idea about. It was a real surprise to us. We didn't set out to do this. Other people had been looking at how sleep evolves and how attributes of sleep are expressed in a very broad range of animals. So there was a very substantial literature, a lot of work through a lot of labs, but in the rather substantial field that examines birdsong learning, because it's such an interesting phenomenon, nobody had really ever considered the possibility. And we essentially, honestly, just stumbled upon it because there was a result that made no sense until it started to make sense.
Yeah, and I'd say that the first question is why study sleep in animals if ultimately we're mostly considered about what's going on in humans. And although we can rigorously study the behavior in humans and, some aspects of underlying mechanisms through like EEG or fMRI, really figuring out what's going on at the circuit and cellular, neuronal, genetic level requires an animal model. So once you need an animal model, then the question is, what animal model are you going to use? And birds provide, surprisingly, a lot of advantages for studying sleep. Birds are an animal species that have profoundly complex vocal learning abilities, vocal production abilities, and the ability to discriminate complex auditory signals, probably better than any other non-human animal in the animal kingdom. And so studying sleep and learning and memory in terms of what birds can do in these specific behaviors that they have is a really useful way to go about it, because you need to have a robust animal model that closely approximates what you're studying. And with birds, with song learning and auditory discrimination abilities, finding out what you can from birds will allow you to generalize to humans better than a lot of other animal species.
Yeah, and what has emerged is really surprising similarities with the description of sleep in mammals. And it's surprising both because the past literature suggested otherwise, and the similarities are at some level actually quite astounding. So it's really turning out to be a very good model both for studying attributes of sleep that are seen in humans and also challenging ideas about how the structure of sleep evolved, and therefore what are the underlying mechanisms.
Maybe you could say a little bit more, Dan, about how similar avian sleep is to human sleep or to mammalian sleep, because you said it's similar, but do you want to expound on that?
Sure. Let me start answering that. And there's quite a bit I can say, of course. And then let me turn it over to Tim, because some of his work has really addressed that quite compellingly as well. So one of the early things that we found in the lab mimicked a seminal set of studies that drove sleep research in hippocampal formation in mammals, which has blossomed into a very major field of study. One of the things we found that was quite similar was there was this phenomenon of neuronal replay. And by replay, what we mean is activity of a neuron that occurs while the animal is sleeping that is very similar to the same pattern that neuron expresses for a particular behavior during the day. And we were recording from a part of the brain that is involved in the production of the song. And so the pattern that we would expect from that is to see the neuron firing in a way that's related to the singing pattern of the bird. And each neuron in this part of the brain has its own particular pattern. And so the experiment involved recording the pattern during the day and then maintaining this microscopic recording over a very long period of time, and then being able to watch while the bird slept. We were able to see that the pattern of the neuron's activity was very, very similar to the pattern that that neuron expressed when the bird was actively singing during the day. And subsequently, we've seen evidence for that pattern of activity in populations of cells. So that really is what motivated the first set of ideas that the phenomenon that we're seeing in birds is related to sleep and is remarkably similar to what had been seen in a very different system in a rodent, but was also described in terms of a replay phenomenon. One of the simplest ways to describe this is when we first described complex sleep patterns in zebrafinches. And we had a poster of this at the big annual Society for Neuroscience. There were images and so on, and there was a little video, and people would walk up and they would just blurt out, "Oh, that's just like my rat." They would just say this spontaneously because it was such a compelling observation. But we can go well beyond that. And I think maybe at this point, Tim could pick up the story.
Yeah, so when talking about bird sleep, it's always important to relate it to human sleep and mammalian sleep. And I'll start with human sleep because that's kind of our baseline. Humans, typically sleep 7 to 9 hours during the night in one consolidated period. And over that, of course, those hours, they typically have sleep cycles that are around 90 minutes that alternate between rapid eye movement sleep and non-rapid eye movement sleep. Early in the night, there's a lot of this non-REM sleep, and specifically a subset of non-REM sleep called slow wave sleep that is characterized by these low frequency, large amplitude waves. Then over time, then transitions into REM sleep. And then you go through multiple of these 90-minute cycles. Early on in the night, there's a lot of slow wave sleep that slowly decreases across the night. Whereas REM sleep starts off early in the night with very little and increases throughout the night. So you have these changing proportions of these different types of sleep that occur. That's a human sleep, a type of mammalian sleep. But there's something like 6,000 mammalian species, and all of them have different types of sleep. And so the most common other mammalian species that are studied are typically rodents, rats or mice, and earlier cats. And even just across cats and rats and humans, the sleep is very different. Cats sleep much longer than humans and rats. Rats, on the other hand, rather than having one long consolidated sleep period, have many bouts of shorter sleep periods. And though rats are nocturnal, they're more active during the night. They still sleep during that period, and though they're less active during the daytime, they're still awake during that period. In contrast, songbirds in particular tend to be more like humans. They're awake during the day and then tend to sleep during the night. This is true of the zebrafinches that Dan mentioned, and also starlings that I study. They also show these patterns of changing slow wave sleep and REM sleep, such that early in the night, there's a significantly more slow wave sleep that decreases across the night. And the REM sleep starts off with relatively low amounts, then increases across the night. So despite being very different species, the sleep patterns and the daily 24-hour structure of activity in birds is remarkably similar to humans. It makes it much easier to compare patterns of performance changes and other aspects of their behavior that is harder to do in rodents.
And to continue with that, as Tim was alluding to, the role of learning in sleep as something that has become, I think, very clearly demonstrated in songbirds. And it turns out to be, therefore, a very useful system to study this central idea that motivates lots of sleep research nowadays. So we know it from behavioral studies, both in juvenile birds learning to sing their song and in adult birds through Tim's own work, studying how birds do perceptual learning of particular songs and how they commit those songs to memory. And of course, it's everybody's experience that sleep is important for learning. It's been difficult to find ways to systematically quantify that and study it and learn about its neural substrates. And so songbirds do offer a particularly good model, and as Tim was saying, a particularly set of compelling observations that are really quite similar to what we see in humans.
It sounds like they are a good model because their sleep is actually quite similar to humans in that they're diurnal and they have the same alternation of REM and non-REM, but also because they have something important that they need to learn, which is very complex and which we can measure with great precision. And so we can actually look at how well they've learned it. So all of those things make them a great model.
There are also a couple of practical reasons, specifically the two species that we were studying in Dan's lab. With zebrafinches, they sing very well in cages. They can live in cages fine, and they can sing very well in cages. Also, the song structure of a zebrafinch is they learn to produce basically a 1-second snippet of song that they produce over and over again, hundreds of times a day, with a very precise stereotypy. So when studying the song of the zebrafinch, you have a very clear output on what the behavior is. So if you do any manipulations, you can see if the song changes, or you can relate whatever neural activity you're trying to record to the specific behavior that's coming out. That makes connecting the brain and the behavior much more reliable. Starlings are another animal that are very capable of living and working in cages, and they're particularly smart. I do these experiments where they initiate trials by probing a port. It produces sounds. They respond to them in one way or another. And being able to perform these tasks and learn these tasks rapidly, it really enables the ability to study both their memory abilities, the formation of their memories, the stabilization or strength of the memories, and additionally, being able to then do sleep recordings with that too, to really, again, connect the learning that they've experienced in the cage with the brain recordings that you're doing during sleep or during the day.
Dan started to talk earlier about replay and about how you have these neurons within the song system of the birds, which, you can really easily measure that they're replaying the kind of song structure during the night. Am I right in thinking that some of those neurons, if you actually listen to the bursting, you can almost hear the song structure? It's that good a match.
A replay isn't always veridical in the sense that it follows precisely the pattern that you'd normally see in the awake animal. And I don't think that represents an error. I think that represents an important signal that is relevant to the sleep-dependent learning process. But because of that, and because different neurons are encoding for different components of the muscular contraction, you need to know something about the language that any one neuron is speaking. So if you want to do it at the level of a single cell and you only have a recording of one cell at a time, you really need to know in advance how that neuron speaks in terms of its bursting patterns to the population of neurons, to the network. If you're recording from a population at once, then yeah, you can do it as is. You could probably do it with the very first recording that you saw because it can be so clear. Now, that's only true in the adult animal. In the juvenile animal, it's a fascinating story, but it's a different story.
Oh, that's so interesting. What you're saying is that if you have a population recording, you can pretty much hear the song from listening to the neural activity. But if you listen to obviously individual neurons, they're just making up small different components of that song. And so it wouldn't be obvious, or it's not always obvious.
Yeah, it's not necessarily that they're making up smaller components, although that's true, but it's also they're encoding a given component in their own unique way. And when you average across all neurons, then it becomes very clear that it's that component as opposed to another one.
And does this replaying of the song happen throughout sleep, or do you have to really look for it?
Both. It's not necessarily easy to see, but it does happen throughout sleep. We actually don't know that as well in adult birds as in juvenile birds, where it clearly occurs both during non-REM and REM sleep. In work that isn't yet published, that is introducing a third species that we're studying, budgies, who aren't songbirds, but are parrots and are part of the sister taxon to songbirds. We're seeing the same phenomenon of complex sleep in those birds, which is one of the questions we were trying to address with that study. And in work that we're beginning to prepare for publication, we believe that the data seem to suggest that the replay is occurring principally during slow wave sleep.
In the adults or just in these budgies?
In adult budgies.
Adult budgies. But you wouldn't say that for starlings, for instance, or, I'm trying to remember the other species that you've worked on.
Zebrafinches.
Zebrafinches, yes.
I would say we don't know that for the other two compellingly. I think what that hints at is that there is a transition from development to adult as to how and when these processes are expressed. I think that's the clearest result so far. But you're absolutely correct. We don't yet know this for a fact for the other species, and therefore it remains an open question.
As far as starlings are concerned, the recordings haven't been done during sleep at individual cell levels to look for replay, so we don't know that replay is happening in starlings. That hasn't been verified. In zebrafinches in the juvenile state, it's difficult to call what's happening during sleep replay because stereotypical pattern hasn't been established yet. What is happening is increased bursting that may be replaying of the developing patterns of behavior or the developing syllables that they're creating. But it's more bursting that's happening during sleep, and it's not necessarily clear if that is replay.
To amplify on the story in the juvenile birds, those experiments were looking at the very first stages of song learning. So for a bird to learn its song, it goes through two processes. One, it hears the songs of other birds, in particular its father, and it commits that song to memory. And then two, it starts to learn to sing. That is, it makes sounds and it listens to its own auditory feedback, and it modifies its vocal output, its singing, based on the feedback it hears. And little juvenile birds produce an early version of song, which is very similar to what we call babbling in our kids as they're learning to speak. It's a very compelling set of observations that's at the heart of what drives the interest in birdsong learning. So we did experiments where we looked at the activity in a particular region of the brain early in development. Just after, in an experimental setup, the bird had just had access to the song of its own species. It had heard it for the very first time. And because of the nature of that experiment, one has to delay a little bit in development when you give the bird the first access to its own song for technical reasons. So during that time, the bird would hear the song and shortly thereafter would start to modify its singing behavior. So those two separate periods that I was just talking about were a little bit conflated because of the nature, the technical constraints of the experiment. So we did the experiment where we exposed the bird to its own song, and the bird heard, and you could even, if you look at the animals, you can tell that they know something really important has happened in their lives. It's like the first time they heard a good zebrafinch song. There are videotapes which show what you just have to call an emotional response of the birds to that. Some birds you'll actually see sort of swoon and actually fall asleep sometimes. It's like it's this powerful thing that's in them. It's quite impressive. So we exposed the birds in a fashion that had been described by Ofer Chernikovsky, a scientist in New York City, who first provided the behavioral evidence for the role of sleep in birdsong learning. And then we scoured all the vocalizations that the animal produced that very first day, looking for any evidence of changes in their singing. And there was none. And yet, if you look the very next morning, already there's clear evidence of the animal changing its singing. So it had committed the songs to memory, but it had yet to start to change its actual song output. And when we recorded on days prior to first being exposed to that song, and on the night and nights following, after the bird had been exposed to the song for the first time, there was already a clear change in the patterns of activity at night after the bird had been exposed to the song, prior to when the bird started to actually change its singing pattern. So those changes we were looking at night were related to a change in the memory state of the animal. And in fact, depending on which tutor song the bird heard, the pattern of change at night was different.
So let me just ask about this pattern of change at night. So what I understood is that you take these juvenile birds being cared for by females who don't sing, and then you expose them to what you call a tutor song. So that's a sample song, which is probably a pretty good one, right? And they have this emotional reaction. And then they continue babbling for the rest of the time they're awake. And then eventually they sleep. And you record from the song system while they're sleeping. And you find these replays, and these replays match the tutor song. And then what you're saying is that the way those replays are configured actually changes across the sleep period. Is that what you meant by changes across sleep?
Approximately, yes. That's the structure of it, and that's the general pattern of observations. But as Tim was saying, what we see early in development, what we see in these young birds, isn't the same sort of activity which is veridical or near veridical replay that you see in adult animals. Instead, what happened was that the neurons in this part of the brain were kind of sluggish before the bird was exposed to song. They were puttering along, but you never saw strong bursting or variation in the pattern of activity like you would see in the adult animal. And then the bird is exposed to the tutor song that amazing day in their lives. And then that night, all of a sudden, the bursting patterns of the neurons has really picked up. They're firing with that high frequency pattern, as Tim was describing, much more frequently. And the patterns and the type of high frequency firing, that's what varies across the groups of birds that were exposed to different tutor songs. So it's actually predictive of the song the bird will sing the next morning, but it's not really the same type of replay that we see in adult songbirds or that's described in adult rats as they're learning a new task in a maze, for instance.
It's not as organized yet because they're still juveniles and they're at the very beginning of the learning curve, I guess.
It maybe is organized for an animal that's babbling, as opposed to for an animal that is, quote, unquote, speaking. So there's much more variation in their vocal output. And so there's correspondingly much more variation and less rigidity in their neuronal representations of that vocal output.
Does it change across the sleep epoch, or does that come later?
There was a hint that there may be a little bit more of it in REM sleep than in slow wave sleep. And the structure of the bursting seems closer to the average pattern in REM sleep than in slow wave sleep, but it was seen clearly in both types of sleep in these very young birds. Another study done by Teresa Nick then followed the patterns of changes over development, also in zebrafinches, and because of the nature of her recordings, she could follow over long periods of time, but couldn't record from individual cells. She saw also very interesting patterns of variation that changed depending on whether she was recording from the animal during the day or at night. So she couldn't really relate it to different attributes of sleep other than the fact that they had some evidence for activity during slow wave sleep, but it was, let's say, incomplete evidence. But it was still very interesting variation that the animal was expressing throughout development. And here's the thing. I'm telling you all this about zebrafinches, and yet it seems a very plausible expectation that something very similar is happening in human children as they're learning speech and language.
Yeah, absolutely. So you have a paper that talks about how this bursting pattern changes across the night, but that might be in slightly older animals. I'm not sure.
Mhm.
Can you expand on that one a little bit?
Yeah, so we had seen this replay phenomenon, and that was interesting. And it was in part of the brain that's involved in learning and maintaining the learned song. But to really support the idea that the replay is related to learning, then in principle, something has to change overnight. You have to see a trace of the changes in the neural activity. And here's a part of our research where we, at least certainly at that time, did not have a technical advantage. We had a technical disadvantage because the way we were doing the experiments, we had to hold the recordings of individual cells over a very long period of time. We're starting to learn now how to move beyond that and do the sort of recordings of populations of cells that are commonly done in rodents, which are really important and a huge technical advantage as well. But in that experiment, what we did was we recorded from individual cells while the birds sang during the day, and then continued, in the best of cases, to hold that recording throughout the night while the birds slept. And for control conditions, we woke up some birds earlier and let other birds sleep longer periods of time. And then in the very best of recordings, we're able to maintain those recordings while the birds sang, even one song, please sing once before we lose the cell, because it wakes up and shakes its head and maybe we lose the cell. But sometimes we didn't lose the cell and we got 1, 2, 3, and in rare occasions, even many songs the following day. And so now you have the activity of the cell prior to the bird going to sleep when it sang, the activity of the cell throughout much of the night, and the activity of the cell after the bird woke up and sang again, and incredibly small data sets that had all of that really well described. It was hard to do. But when we did that, what we saw was that there was a clear pattern where the cells would tend to change in a systematic way over the night. And most cells that we were able to do these recordings for showed that same pattern of change. And this is in the part of the brain that is actually driving the last stage in the forebrain that is sending commands to the brainstem that's driving singing. So all those cells in that part of the brain are changing, and yet the song is changing microscopically at most. And so those changes can't be occurring at random. They have to be changing in a systematic, coordinated fashion. And that suggests information processing. It's not just random changes that are occurring, but it must be some sort of structured change.
But there was no clear relationship between improvements in the song and how much the changes had occurred.
There's other evidence that shows that when the song changes in the adult, the neurons change also. But in the experiment we're talking about now, we weren't able to relate directly the changes in the patterns of the neurons with the changes in the behavior of the animal.
And it could be because these were adults instead of juveniles.
I think the reason is that we're right at the edge of being able to detect all of these changes. What we prefer to do is to modify the song and see if the neurons change. And an experiment was done like that. And although it's interesting, they didn't much focus on it, one very clear result was that the part of the brain that's called the basal ganglia, which exists in birds and exists in mammals and perhaps exists in all vertebrates. And it's important for speech initiation, language initiation in humans. That part of the brain showed changes in activity when the bird was conditioned to change its singing pattern. So all that is about how the bird produces its song. There's a whole other side to this, which at some point I want to emphasize that I hope we get to, which is how the bird hears and remembers its song.
We will definitely get to it. But let me just ask a couple more things about this. So you mentioned that you think these changes across sleep index a kind of processing. What do you think that processing is doing? What do you think is actually happening?
It's a great question, and it's also an impossible question, because there isn't any one thing that sleep does, right? Wouldn't you agree with that? You can't really say what sleep is for in humans.
Absolutely.
it's for 10 or 10,000 things. Right? And I think that that's true also in songbirds and in birdsong learning, because if I were to ask you, is birdsong learning represents the type of memory which we call procedural memory, where you learn to do some pattern, say some sequence of things, you'd go, yeah, it's like procedural learning. And then if I said, well, is birdsong learning like where you learn the names of different objects and how they're related to each other, which is what we call declarative learning, you'd pause and you'd go, yeah, it certainly has attributes of declarative learning. And we could go on and on and on. Birdsong learning is a whole organism phenomenon, and it involves motor learning and sensory motor learning and procedural and declarative learning and all these things. And sleep is involved in all those aspects, I believe, and I think the emerging evidence strongly supports that. So I have ideas. In particular, I think the reconsolidation phenomenon that Tim was central in demonstrating in birds and studied to tremendous advantage, I think there's some very clear ideas that emerge from that, but we'd first have to talk about reconsolidation to talk about that.
We will definitely come to the reconsolidation. Let me just ask you about one more part of this story before we move to the listening part. One that was, I thought, very important, De Regna Court 2005, where they had some very interesting findings about how the extent to which the song changes overnight, I think, related to the replay. Would you mind expanding on that a little bit?
I would be happy, and I'll start with a sort of an amusing anecdote. I was trained as a neuroethologist, and so what that means is we take advantage of animal behavior viewed in naturalistic conditions, but really perceived of through the process of evolution and evolutionary change, and use those insights in animal behavior to guide our questions about mechanisms. And there we were studying this phenomenon, the replay phenomenon that we talked about, but at the same time suggested that sleep was important for birdsong learning. And what a crazy idea that was, because nobody had ever talked about that before. It's a little peculiar when the electrophysiology suggests the behavior in contrast to studying the behavior and using that to guide the electrophysiology. So when Ofer Chernikovsky and his students published that paper, it was both exciting for us and for me more than a sigh of relief to know that we weren't completely crazy in what we had been saying. So what did they do? They developed first a set of techniques that allowed them to record and follow the process of song learning over the entire song learning period. So one bird might produce as many as 1 million different song snippets, and they recorded them all. And then they had techniques that allowed them to start from the adult song and to work backwards in time to figure out what the classification of the different snippets of songs that appeared very early in song development. And in a previous paper where they had done that, they had seen some evidence that there was some structure to changes in song that maybe were related to sleep. So then they designed a series of experiments to investigate that. As in the other experiments, they arranged an apparatus of birds. They could control when birds were exposed to songs and how many songs they were exposed to. And they took juvenile zebrafinches and exposed them to a minimal number of songs on one day and then each of many subsequent days. And they recorded their singing on days prior to that exposure to song and on days after that exposure to song and throughout all the way to adulthood. And what they found was that initially the patterns of singing behavior didn't fluctuate very much. But then when the birds were exposed to the song, starting on the very next day, there was a dramatic change in how the birds sang. And there was variation in the quality of the song that the birds sang if you compared it to the target song, the tutor song that the birds were trying to copy. So initially in the morning, each bird sang a rather jumbled up song that wasn't very well structured. And then by the afternoon, the song was already starting to show more structure. So that was evidence of learning. And by more structure, what I mean is more similar to the target song that the bird was trying to match. But then, remarkably, the bird would go to sleep, and the next morning its song would be jumbled up again. And then it would improve again in the afternoon, and the birds would continue to iterate that day after day after day. Now, you may think that that's counterproductive, but it's not. Because one thing they showed was that the birds that showed larger variation in those patterns were the ones that learned the best. And they also showed that this was directly related to sleep by either not allowing birds to sleep in the morning and then seeing if the same phenomenon was expressed later in the day when the birds first started to sleep, and it was. So that means it wasn't tied just to the time of day, like some aspect of the circadian pattern. It was specifically tied to sleep. And they also put birds to sleep by giving them melatonin, which admittedly might have reset lots of rhythms in the bird. But when they put the birds to sleep with melatonin during the day, and then the birds woke up later in the day, they also reiterated that pattern. So the totality of all these results suggests that there's a rhythm that the birds express, which is counterintuitive: more babbling in the morning, learning, and then, quote unquote, losing that learning and more babbling again. I think it actually makes sense, though. I think there's an explanation for what's going on, but I'll let you ask the question. That's
so
correct.
What's the explanation for that?
Again, this is not just about one type of learning. This is about whole animal learning. There are two things going on. The animal is committing a song to memory, and the very first time he hears that song, he doesn't have a perfect copy of that. And we know that because we know that early when the animal is committing a song to memory, early in development, that memory can be changed if the animal, for instance, hears a different song. So that memory has to be reinforced over time. So there's that dynamic going on in the perceptual system. And there's also a dynamic going on in the production system, and the two are interacting with each other. One hypothesis is that the animal isn't getting feedback at night, and so his motor system is drifting. And so when he wakes up the following morning, he's now off somewhere, and it takes him a while to recover from that drift and get back to his normal song. That's one hypothesis. But I think another hypothesis, which I personally favor, and I think the subsequent evidence maybe tends to support, is that there's an interaction between the two. You might think of it as a top-down and bottom-up interaction, bottom-up being the perceptual system feeding up into the higher centers of the brain, and the top-down being the higher centers of the brain feeding down to the motor system and feedback through the perceptual system. And in fact, there's even a very interesting algorithm called the sleep-wake algorithm, which was described by Jeff Hinton, which is a computational framework for thinking about these sorts of interactions. And I think that conceptualization of it is a very plausible explanation for why you see this dramatic variation in the birds, which is driven explicitly by sleep-dependent learning.
So this processing that we've said earlier is happening during sleep has to be part of this story, right? So maybe that processing is involved in the interaction that you just described, or in addition to, it's somehow the tearing apart of the memory, is the kind of processing that has to happen maybe in order to allow these two parts to integrate better. That's kind of, yeah.
That's what I'm thinking. I'm thinking the animal is improving his motor memory as he's practicing during the day. And at night, perhaps, there's an interaction between that motor memory and the sensory memory that the animal has committed during the day. And that sort of pushes him in a somewhat different direction. Because initially, in my conceptualization of it, the two systems are not well coordinated with each other. They haven't tuned each other up. So there's sort of a push-pull relationship between the two of them. And so the animal is in a different state when he wakes up in the morning than when he went to sleep at night. And so now when the motor system comes along and goes, okay, let's go and sing again, all of a sudden it finds that when it tries to hit an A, it's missing the A and it's hitting some other note or some other pattern of notes, really. And so it has to work at it to get back to that pattern of notes and do a better job of hitting the A's and the sharp notes and so on and so forth. And so it improves over the day, and then it goes to sleep, and now the system underneath it changes it without the higher levels are necessarily coordinating that. But then over a period of time, the two systems become coordinated, and that is what the learning phenomenon represents.
Yeah, to me, it makes perfect sense. And I see what you mean about the Hinton sleep-wake algorithm as well. I think that's a kind of a backprop error-driven learning algorithm. So it would be like, basically, you've trained something and it's producing an output, and that's all happened during the day. And now during sleep, you get a chance to check how good that output is against your reference, which would be the sensory thing, and say, well, these are the errors you're making. We need to adjust everything. So now everything is going to not work so well for a while until you retrain it again.
You could ask why this takes so long. I mean, for a little songbird committing 2 months of its life, early in life, to learning to sing, from an evolutionary perspective, that's a huge commitment. So why? Song evolves under sexual selection. So in many species, males sing and females choose males in part on the basis of this characteristic singing. If the male isn't singing well, forget about it. So they have to be experts, real experts. And so they have to learn slowly. So if you learn quickly, you can get an approximation, but you can't do a great job.
Yeah, it's great. I think it's such exciting work, such exciting ideas. We are actually trying to build a model at the moment of not birds specifically, but a computational model that takes into account replay and sleep and what it might do for consolidation. I don't think it will be quite as good as what you've just described, though.
Well, let's keep talking. Maybe we can make it better.
Maybe, yeah.
I'd be happy to. Yeah.
So great. So, I mean, that's a wonderful story, but you alluded earlier to some of your work and also with Tim on how birds listen to song and how they learn to do that and how that consolidates. So maybe we can move now into talking about that stuff. Did this all start up when Tim came to your lab?
Yeah, so the zebrafinches are studied primarily for the vocal production, the song learning abilities of them. There's a lot of work on the auditory side of that too, especially because, as Dan just mentioned, the females listen very acutely to the song that the males are producing. They have to be able to discriminate very precise differences in singing. But starlings are not nearly as good for studying vocal production because their songs are so much more complex. Rather than learning about a 1-second motif of song that they repeat over and over again, starlings develop a collection of motifs that are, again, about 1 second complex vocalizations of songs. But they, in the wild, will, over their lives, continue to add to it to the point where each starling may have 50 or 100 different motifs that it sings. And each starling has a unique set of these motifs. Their song bouts are consisted of stringing together motifs in somewhat of a sequence of the particular type of motifs that they use. But in any case, every song rendition is going to be different. And every starling has a different group of motifs that they sing from. So other starlings can identify starlings based on the songs that they sing. Their auditory memory abilities are really impressive, given that starlings are very social creatures and live in groups of tens of thousands. So starlings are very good at differentiating and remembering these complex motifs. And from that, I was coming to Dan's lab from the human side of sleep and memory. On the human side, the behavior is studied so well, we can bring humans into a lab, train them on almost anything, then have them come back later, either after a full day awake or after a night of sleep, to see how the memory has changed. To do something similar in animals, you need to be able to train the animal on something in a very short period of time that then allows you to then retest it after, say, a full day awake or a night of sleep. So I did this in starlings. I would train them to discriminate between 5-second segments of natural starling song. So they'd be in their cage, they'd initiate a trial. One of two sounds would come back, one of two different 5-second segments of song, and they would learn to respond to it in what's called a go, no-go procedure. So for one of the sounds, they would respond again and they would get a food reward. And for the other sound, if they responded, the lights in the cage would go out, which would indicate that that was the wrong response and they couldn't run another trial for about 10 seconds. So over the course of responding to these sounds, they would learn, oh, I want to respond to this sound and I don't want to respond to that sound. And that's how we would measure whether or not they could discriminate between the sounds. And so initially, I would give them about a 2-hour training period of discriminating between these sounds, and they would show significantly greater performance than chant, showing that they had learned to discriminate these sounds.
How could you tell they could discriminate? So they're hearing two different snippets of song, and what do they do to show you that they can tell the difference?
Yeah, so they're in their cage. There's an apparatus that has a hole in it that they can stick their beak into, and then there's an infrared sensor behind that registers if the beak has broken the plane. So they run the trials on their own pace. They initiate a trial, then one of the two sounds plays back. And then they will either respond to it or not respond to it. They have 2 seconds to stick their beak back into the port to break the infrared plane to show that they responded. So these two sounds aren't playing at the same time. Just over the course of running mini trials, they will hear the two different sounds and learn that when they hear one of the sounds, they want to respond and they'll get a food reward. And when they hear the other sound, they want to withhold their response. Mm-hmm. And then they can just run another trial again in a couple seconds rather than having the lights in the cage go out and them being prevented from running another trial again.
So they learned either to respond positively to one song or withhold their response to another song. And they learned to do that just within a couple of hours of exposure to these songs.
So they learned to recognize these songs. And so now what you want to investigate is whether their ability to do that changes across sleep, right? So can you tell me about how you tested that?
Yeah, so I took the standard design that is how this is studied in humans and applied it to starlings. And this is a very specific case where the starling wake-sleep pattern of being awake during the day and asleep at night, similar to humans, really is important because I can train them in the morning or I can train them in the evening and then retest them 12 hours later that evening or 12 hours later the following morning and look at how the performance changes across either a period that consisted only of wakefulness or that included a full night of sleep. And when we did this in the initial study, over the course of the day, when they were retested in the evening, their performance was a little bit lower, not significantly so, but just a little bit lower than where they were in the morning. But if we trained them in the evening and retested in the morning, their performance was significantly better, in many ways replicating the type of enhancement effect that had been shown in some human work. Looking only at the 12-hour conditions, there are circadian issues that could explain those effects because the training and the retesting take place at different times of day. So if the performance is naturally, say, better in the morning or naturally better in the evening, you'll get changes that aren't related to consolidation, but are more about circadian factors. So we also use 24-hour groups where the animals are trained in the morning, then retested 24 hours later after a day awake and a night of sleep, or in the evening after a night of sleep and then a day awake. And in both cases include sleep, and in both cases, performance was significantly better after that 24-hour period. We then replicated in this where in the same birds, they would be tested after a full day awake and then after a full night of sleep, or trained in the evening and retested in the morning and then the next evening. In each case, if the starlings had sleep during their period, their performance on the auditory classification task was significantly better. Whereas if there was only wakefulness, performance was about the same or slightly worse than it was after the training session. That was really the first example, especially in an animal, that used the methods that have been used in humans to show the sleep-dependent memory benefits. And it was showing essentially the same pattern in starlings.
It's really amazing that it shows exactly the same pattern as had been shown in humans.
Not only Tim demonstrated that in the initial study with starlings, but he also was thinking about that result in terms of another set of results in the human literature that was very puzzling to us and that was in some sense inconsistent with his result in starlings, and he pursued that work as well. So I wonder, Tim, if you would be willing to talk about that.
So that actually relates to this follow-up study that I did in the starlings. And what this has to do with was the different patterns that we were seeing across human and bird results. In the human research, the pattern that people often found was that performance remained stable across the day and then was better after a night of sleep, was significantly higher than the post-training levels. Yet in a study that Dan did with our collaborator Howard Nussbaum and Kim Fenn was the lead author, in a study of speech perceptual learning, what they found was that performance actually got significantly worse over the day and then recovered after a night of sleep. And so there is this question about whether it depends on the type of task as to what the pattern is, whether memory gets worse over the day or becomes stabilized over the day, or whether memories actually are enhanced after a night of sleep. So the first study that I did with Dan and with Howard was actually looking at first-person shooter video game learning. And it also showed a pattern where performance got worse over the day and then recovered after a night of sleep. So at the time, we thought maybe these are more complex tasks rather than, say, this very rote learning of just typing a sequence over and over again, as was the most common motor sequence learning task in the sleep field. When you looked at the individual key presses in this motor sequence task, over the course of these 30-second trials where people were typing a sequence as quickly as they could, in the last half of it or last 10 seconds, the key presses would slow down. The task is asking you to type these sequences as quickly as you can, so it's kind of like a sprint. But by the end of that sprint, you were tiring out. And so what happened was when you looked at the final training trials, which were considered the level that you were comparing to your post-training levels, they were actually lower than the real training level. They were confounded by this fatigue that was building up during the training session. And so we did a couple of different manipulations that involved either just giving the participants a 5-minute rest to get over that fatigue or to include more rest during the training. And what you found was that the performance at the end of training or at the end of that 5-minute rest period was significantly higher than it had been just 5 minutes earlier. Or if you gave more rest during the training practices, you didn't need a 5-minute rest because you never experienced that fatigue. And so ultimately, when you use this new higher value as a more accurate representation of what was learned during the task, you find that performance on the motor sequence task also decreased during the day, significantly so, and then recovered after a night of sleep. So it seems that at least across these varying tasks, one video game learning, one in speech perception learning, and in motor sequence typing, that performance would get worse over the day and then recover. Question is, one, why does that happen? And then two, as far as the Starlings went, we didn't see that in the Starlings. Starlings' performance remained stable and then got better over sleep. And so we thought this might be related to interference. In human studies, you go into the lab, you do the study in the morning, then you go about your day doing whatever you do, and then you come back in the evening. And so there's 12 hours worth of uncontrolled potential interference going on that may be contributing to why performance gets a little bit worse over the day. In the Starlings, there wasn't such interference. The Starlings lived alone in a cage, and throughout the day, they only experienced one very familiar stimulus. So there was essentially no other learning going on, nothing else happening. Again, looking at the motor sequence field and work by Matt Walker that extended learning one sequence to looking at what happens when you learn two sequences, I sort of implemented an interference study with the Starlings, where now what happens when the Starlings have to learn to discriminate between first two stimuli. So they train on that for a couple of hours, and then they have to learn a different discrimination of two more songs. So now they've had to learn 4 different sets of stimuli across the day. And when they're retested in the evening, their performance is significantly worse. Yet, when the Starlings are retested the following morning, their performance has recovered. And so it seems that learning multiple tasks across the day prior to sleep is producing competition between the memories, but whatever's going on during sleep is sort of separating that out. And performance after sleep is as good as if there was never that interference in the first place.
Can I check if I got it? So you described 2 experiments here. The first one, basically, you showed that performance on this finger tapping task in humans deteriorates across a day of going about their normal activities, and it improves again across sleep. So that's consistent with the literature on finger tapping that's already there.
Yeah, I mean, that was a significant reinterpretation.
Like at that point, the main hypothesis that was being proposed was that memories are stabilized across wakefulness and then enhanced after sleep. This was showing that memories were actually getting worse during the day and then recovering or getting enhanced after sleep.
I mean, specifically, Reichardt made the first challenge to that and suggested the fatigue hypothesis, but he never actually did the sleep study. That was what you would have to do to test that.
He did a small amount of the sleep study, just not enough to fully.
Okay, I don't remember that. And then Tim came and said, look, this has to be done right. And he went ahead and did the full study and got the result that he was just describing.
So let me see again if I got it. So Reichardt proposed that the kinds of sleep effects which Matt Walker had found on this finger tapping task were just due to fatigue. So basically when people train, they get tired, and that's why they're better the next morning. Now, you controlled for this, as I understood it, by allowing people to have a little 5-minute rest at the end, and then you tested them again after this rest. And did you do that both in the morning and in the evening, and then you used that as your actual measure? Is that what you did?
Yes, there were 4 versions of it. There was the way that Matt Walker had done it, which was with 30-second trials followed by 30 seconds of rest. I did that version and then added a 5-minute rest period at the end of it to see what changed over that 5-minute period. And performance got much better over that 5-minute period. And then when you looked at the speed of the tapping, you could see that after the 5-minute rest, that fatigue had disappeared.
So then you use that measure after the 5-minute rest as your measure. Yes.
Yes.
Okay.
But then I also did the Rickard manipulation, which was to, rather than do 30-second trials followed by 30 seconds of rest, he did 10-second trials followed by 30 seconds of rest. So that just makes the sequences shorter so you don't develop fatigue in the same way. And in that case, you don't need a 5-minute rest period. Your performance is already at a higher level because fatigue was never weakening your performance.
So with this kind of combination of things, you were able to separate out the impact of fatigue from this interference that's happening across the day and say that these are two separate things, basically.
Well, once you could correct for the fatigue, the performance level was higher. So that when the performance was then retested at the end of the day, it was relatively lower compared to this higher and more accurate post-training performance level.
Right.
Yeah.
Right. So they got worse across the day, even if you gave them this 5 minutes of recuperation time at the end.
Yes.
Okay, got it. So that was the first experiment you described. And now the second one, you moved on and you thought, okay, they're getting worse across the day. Maybe this is because there's interference happening. So now I'm going to design an experiment which directly tests that hypothesis by training them first on one sequence and then on an interfering sequence and checking what that does to performance.
Well, so now this is switching back to the starlings, where we had just shown that when they only learn one discrimination during the day, their performance does remain stable or just very slightly worse. And now if we train them on 2 different discriminations across the day, when they get retested on that first one, their performance is now significantly worse, showing that having learned that second discrimination has impaired their ability to remember the first discrimination. And so now the performance loss across the day is looking similar to what is found in humans. And so that's called retroactive interference, where the second memory that you learned interferes with the first memory. But it also works the other way in proactive interference. So if we, instead of testing on the first memory that we trained, if we test on the second one at the end of the day, that performance is also significantly worse. So over the course of the day, these 2 memories that they've learned are interfering with each other such that they are both worse later in the day.
You see the amazing connection between the research results in these 2 fields and how one has driven the other. I think that's both really exciting, and it really makes a strong argument that the connection isn't superficial, but it's quite deep. And so at some level, the patterns that we see in the behaviors and the patterns that we see in the mechanisms driving those behaviors have to share some similarities.
Yeah, I completely agree. I mean, I think the synergy is really exciting. However, I do have one question about the synergy, which is before we move to talking about the listening studies, we were talking about this idea that the learning needs to get worse across sleep because you're trying to integrate 2 things together so that the behavior will actually be worse. But now in these studies, we're seeing that the behavior is getting better across sleep. Mm-hmm. So how do we reconcile these findings?
In my view, one is a perceptual task. The other one is a sensory motor task. And so I think the behavior gets worse when you have an interaction between 2 systems that are initially not well coordinated with each other, as you would have during perceptual learning and motor learning in a juvenile organism, be it a human or a songbird or whatever. But in the adult animal, when you're looking at perceptual learning, there might be a different set of rules.
Okay, that makes a lot of sense. So it's when you need to integrate perceptual and behavioral that you might get the opposite pattern.
I'd say that I think that also has to do with the fact that in the juvenile zebrafinches, it's a system of learning a memory that takes 2 months to learn. And it's this incredibly important behavior that is very evolutionarily inspired. Whereas, say, in the starlings, they're learning a discrimination that they could probably figure out in 30 minutes or something, maybe less. Consolidating a simple memory versus something that is this incredibly long, drawn-out process is going to have clearer effects. And then also for a process such as vocal learning, where they're trying to ultimately produce a very precise output that starts off with very imprecise output, you don't want to consolidate an incorrect memory. If they were consolidating and stabilizing early productions, they'd end up producing a very bad song. And so there might be developmental issues as to why you might not consolidate in the same way.
And so there's probably huge impacts of all the neuroplasticity and pruning and brain development and other things that are happening in that juvenile bird brain at the same time, right?
Definitely.
Okay, so far it's already a fascinating story, but I know that what you did then, Tim, was go on and do something that's arguably even more fascinating and relates to reconsolidation. So could you maybe explain that one?
Yeah. So in the study of sleep and memory, the original idea for memory consolidation that was after a memory is encoded, it's going to go through these processes that ultimately lead to a stable memory. But we know that memories change and we don't always have perfect access to them over time. And so this idea of reconsolidation came around that actually memories cycle between stable states and more labile states where they can be affected by other things that are going on. They can be changed or updated. And it's thought that what brings a memory that has been consolidated and is in a stable state back to being labile is simply the activation of that memory or recalling that memory. And by recalling the memory, processes are brought in such that new experiences or whatever is going on can affect that memory. And so with the first two studies I'd done, I looked at just what happens over the first night. So day 1, day 2. The reconsolidation study was really to see, well, what happens over multiple days? Does sleep continue to benefit the memory? And what happens when we start putting in these interfering experiences over the course of multiple days? So instead of looking at just the day 1 and day 2, we first looked at the day 3, and then we did a 5-day experiment. And so we trained the starling, as I had done before, in the morning on day 1, and then retested it that evening, and then retested it again the following morning and evening for the next 5 days to see how the memory was changing. And then for 2 of those conditions, on the subsequent days, on days 2, 3, and 4, we also included new interfering experiences. So on day 2, there was a second song pair that they had to learn. On day 3, there was a third pair. And on day 4, there was a fourth pair. We wanted to test, one, what happens on additional nights of sleep. And initially what we found, when the starling only learned one song, performance didn't get significantly better after the second night. So the first night, there was a big improvement, but the nights thereafter, performance essentially just got incrementally better, which was expected since each testing experience is a little bit of a training experience. The 2 interference conditions were the real question about what we wanted to look for in terms of reconsolidation. So in one condition, we trained them on the interfering task before they had been retested on the original task A that we were testing throughout the whole experience. And the reason we tested it before was because the idea in reconsolidation is that by utilizing the memory or recalling the memory, that makes it active again. So if we train them before they've recalled the memory should remain stable. But if we train the interfering experience after they've recalled the memory, after they've been retested on it, then that memory has been activated and is thought to be in a labile state where now an interfering experience can affect it. And essentially, that's what we found. Over the 5-day period, when the interfering experience was encountered in the afternoon after they had been retested on the original memory, performance on the original memory was then significantly worse in the evening, but then recovered and was enhanced after a night of sleep. So you'd see this pattern where over the course of the day, they'd get better over the first night of sleep, then they'd get an interfering experience, and then performance would be worse after that in the evening. And then they'd get better over the course of the sleep, and then they'd get another interfering experience, and then test it again later on the 3rd day, and that would get worse again. So you'd see this cycle of when the interfering experience happened after the memory had been reactivated, it interfered with it. But if the interfering experience occurred before the memory had been activated that day, it didn't produce that interference. In any case, what you would see is that by activating the memory, it seemed to bring it back to a labile state, allowing it to be changed. But then there's these sleep-dependent memory benefits that you didn't see in the case of the control condition that didn't have these interfering experiences at all.
So you train them on one song recognition, and then you might present your interfering new training right after you tested them. So you've reactivated by testing, and the idea is that that's made the memory labile. Whereas if you didn't reactivate, so if you never tested them in the morning, but you just presented a new training, it wouldn't be labile. And so you only get interference if you first test on the thing they already learned, and then you train them on something new. And you basically show that if you do it again the next day, it repeats again.
Exactly. Yeah. And we also did then further studies, which is what you were suggesting. Rather than testing the original memory on each day in between, we only presented the interfering experiences and then just did the original memory test at the end of the experiment. And what we ended up finding was that in both cases, whether there was interfering experiences during those 3 days or whether they learned 3 new tasks during those 3 days, performance on the original memory on day 5 showed the significant memory benefits, that learning these 3 interfering experiences didn't impair their ability to remember the initial one. And in fact, the data suggested that having trained on these additional interfering experiences may have even enhanced the memory of the original learning.
So that's what I remember from this paper, by the way. I remember the really exciting thing for me was this idea that you'll always get a performance improvement across the first night. But if there's no interference, you might not get a performance improvement across subsequent nights. So the second night, third night, whatever. But if you present this interference, so they learn this other thing on day 2, then you get a performance improvement again across the subsequent night. And so what I took from that, and tell me that if this is wrong, but what I took from that is you were suggesting that by giving something new to consolidate, so not just the original sequence, you're actually kind of kickstarting the consolidation process again. So now both sequences, the new one and the old one, are going to get worked on during that next night of sleep.
Yes, that's pretty much exactly the idea that I was trying to lay out there, especially in the sense where the original memory is trained on day 1 and retested on day 5. And now you've thrown in 3 different whole learning experiences. At the very least, you would expect that maybe they aren't going to remember the original memory as much, but not only did they remember it as well, they probably remembered it significantly better. Work that I'm working on right now that has done EEG recordings in starlings during the learning process, what we find is that on the night of sleep after training, there appears to be a pretty substantial increase in the amount of slow wave sleep that occurs very early in the night. And that correlates pretty strongly with the overall performance improvements. We don't have strong mechanistic understanding what's going there, but the time course of how that's working and the fact that it's slow wave sleep early in the night is very consistent with both rodent and human models. In this case where there's 3 days in between related learning, my suggestion is that on night 1, there's the original consolidation. Slow wave sleep is probably increased and correlates with that improvement that occurs over the first day. But now when you throw in a different task on day 2, that's going to instantiate a night of consolidation for that second set of stimuli. But because that is related to the initial one, it's going to cause a reactivation or also benefit the recently learned memories. And there's other unpublished work in Dan's lab that has shown that when you train an animal on one of these song pairs for a while and then switch to another one, it causes a reactivation of that original one. So I think that each night that different interfering experience was happening, that was causing an increase in slow wave sleep that was at least tangentially benefiting the original memory. And so that would be a way to use other similar learning experiences to actually enhance a related experience without additional training.
Oh, that's very exciting. So you have the EEG recordings from both nights, the first and the second night. And in both cases, you get an increase in slow wave.
This was going more back to the first 2 experiments that only looked at the training day and then the post-training day.
So your hypothesis is that if you did an experiment where you had the EEG on both nights, you would always find an increase in slow wave sleep on the first night, and that's what you've seen. But you might find that you only get an increase in slow wave sleep on the second night if you learned a new task on day 2, basically.
That would be my guess.
You should do that. That would be so interesting.
Yeah, Tim, you should do that. Come back to the lab and do that.
You suggested this as a mechanism for boosting learning in general, basically by saying if you keep learning new things, then it should boost other things that you've learned previously. So it'll be a synergistic ongoing process.
Yes.
That's really novel. I don't think I've heard that. Have you heard other people talking about this, or is this a brand new idea?
I haven't heard other people talking about it.
I guess it raises questions about how general the sleep consolidation mechanism is. Some people would argue that certain memories will be tagged for consolidation, and those ones will get strengthened and others won't be worked on. It may depend on the scenario and the task. I don't know what you guys think about that.
So how do they get tagged?
Well, one idea is salience, that things which are important, emotional, arousing will be preferentially consolidated. But another thing is if we trigger them for replay or things that were learned more recently or things that haven't been consolidated beyond a certain point, those may all get consolidated as well. So what you're suggesting is basically if we can make the memory labile, then that pushes it into that state where it's going to get worked on during sleep.
Yeah. And also, again, there are all these boundary conditions on it. I don't know if we did the initial training and then say a week later, a month later, interference training, I imagine at that point it would no longer be susceptible to being affected by, say, learning much later. So it may have to do with the fact that these are all learned within a relatively close period of time. Another issue is that these memories within the starling auditory system are represented by overlapping distributions of cells. So if similar sets of neurons are involved in the memory for discriminating the initial song pairs, and if an overlapping set of those cells are also involved in learning the interfering cells, when sleep consolidation mechanisms are consolidating the interfering experiences, that might naturally benefit the recently learned memories that are also represented by that similar set of cells.
I think that it's worth thinking about the problem that nervous systems have to solve. If we sort of take a step back and consider the problem more generally, this fits in very well. So exactly as Tim was saying, nervous systems have a problem where inputs that are activating very similar populations of cells in very similar patterns of activity have to be resolved one from the other. And one of the ways they do that is to build up in perhaps something of a hierarchical fashion, representations that are more dissimilar from each other as you move up in the nervous system. So whereas, say, in the ear, if you look at the patterns of activity going to the first stage of the auditory brainstem, it's quite difficult. It can be done, but especially for complex sounds, it would be very difficult to understand one population of cells hearing one song from the same population of cells hearing a different song. If you go up into the auditory cortex or higher levels of the auditory cortex beyond the primary auditory cortex, you might find individual cells that start to make this distinction. But nevertheless, you now have to represent those patterns in long-term memories. And so the nervous system's job in general is to enhance the contrast between when there's variation in the external world, to tend to enhance that contrast in the patterns of populations of cells. So at very fundamental levels of sensory systems, there's a phenomenon called lateral inhibition, where cells that are close to each other physically in the brain tend to inhibit each other. And that phenomenon leads to the ability to detect edges or sudden changes in contrast in sensory systems. I think what we're talking about here is the same phenomenon of lateral inhibition, but in a much higher dimensional space. I think what the brain is doing the same thing. It's saying, this song is different from those other songs, and I have to represent it differently, and I have to figure out where the edge is, where the boundary is, so I can say, that's this song, and that other thing is that song. And so I'm building up a pattern of, think of it as sort of lateral inhibition, but more complex version of it, more dimensions, so I can say, that's one memory and that's another memory. And so if another song comes along which is similar enough, it's going to activate that memory. And activating that older memory, still a new memory, but one that I've already made, will help me figure out how to fit those different memories into the same space that I've got for song memories. And so it's something along those lines, I think, that's happening. And what Tim's work has done is provided both insight into that process and an experimental paradigm to study it at the level of single cells and populations of cells.
And it also relates very nicely to what we were saying before about if you have to integrate together slightly different sets of information, then maybe you need to work on that during sleep.
Yep.
Has anyone managed to record the replay in conjunction with the behavioral tasks? I know you said you can't do it easily in Starlings.
No one has, and that mainly means I haven't. Originally, the intention was after the first study that we did showing that sleep benefited memory in Starlings, my plan was then to start doing single cell physiology. And just as a coincidence, Dan's lab was particularly full at the time, and we didn't have data acquisition systems for me. So I had to wait a few months before it was going to become available. And in that meantime, I started doing the interference studies, which then just sort of took on a whole life of their own. And I never got back around to doing the full physiology. But I think the entire time that I was doing the work in starlings, and still to this day, I have a lot of ideas about how to look for replay in starlings and to see both the locations where the memories might be and to be able to relate it to the behavior, because the behavior in the starlings is both quite stable and pretty robust.
Yeah, and I think now we have technologies at our fingertips that we didn't have before, which would actually make the experiments a whole lot easier. So I think those are just great experiments waiting to happen.
So plenty of fodder for future work there. Yeah, it's wonderful. So I'm conscious that we probably should start to wrap up. I've taken an awful lot of your time already. Let me just ask, is there anything else I should be asking you about?
I think we've covered a lot of ground. For me, the behavior in the animals, of course, has always been the central motivating factor to drive the research. But it's incredibly satisfying to see it connect in so many ways and so compellingly with behavior in other animals, especially behavior in humans. This grew out of an interaction initially with my wonderful colleague in psychology here, Howard Nussbaum, and then students who came through in psychology initially, like Tim and others who have followed. And those connections just develop and become stronger and stronger. It's very satisfying to see that. It really gives one a sense that the neuroethological approach can be applied with great success to studying humans, and that studies in humans can tell us the next experiment to do in songbirds. Isn't that cool?
Yeah, it's really a great model system for studying sleep and memory.
You mean humans? Yes, I agree.
Yes. Okay, wonderful. Well, so on that note, let's finish. It's been really wonderful, and I learned a lot, and I really enjoyed hearing your stories. You've been listening to the Sleep Science Podcast with me, Penny Lewis. My guests today were Professor Dan Margoliash from the University of Chicago and Dr. Tim Brawn from MIT. Our producer was Dominic Carr. If you've enjoyed the show, please consider subscribing to the podcast or liking us on Twitter. We're planning a Q&A session for the last episode of this series, so if you have questions about this episode, any other episode, or anything at all sleep-related, please send them to us at #SleepSciencePodcastQ&A. Thanks for listening, and until next time, sleep well.
Oh, and I have one more thing. You didn't comment on my shirt.
I've actually just been staring at it. Thinking like, is it supposed to be, at first I was thinking dice. Now I'm thinking paint splotches. What is it?
These are bird droppings from different species.
Oh, right. Like they're predictably different shapes like that. Which one is the starling?
You can't see it, but it has in black here the names of any number of different species. I have no idea what the starling is. All of them at once. It's an incredibly messy bird in the lab.
I need to get a shirt like that for my PhD supervisor, Alex Koselnik, who you might know. Yeah. He works on starlings.
I didn't know that he was your PhD advisor.
Yeah.
Really?
Yeah.
Well, I want to hear that story sometime. That's interesting.